much for this great opportunity. And I'm really set up so terrifically by Dr. Soan's prior, because that was, and maybe we can get us on time, but maybe not. So with that, just to make sure I can advance my slides, here are my disclosures. My current disclosures actually relate to PARDS phenotype development, which are funded by the National Institute of Health right now. And I think since we learned about pathophysiology of ARDS and the history of ARDS with the prior two excellent talks, I wanted to take a step back, since we're focusing on biomarkers, to make sure everybody has a common framework for biomarkers. So the WHO more than 20 years ago identified a biomarker as not just a substance or structure, but it's something that does something important, right? It can influence or predict the incidence or outcome of disease. So just because you can measure it might be a good thing, but it needs to have a relevance. Likewise, within the US, the NIH and the Federal Drug Administration 6 years ago identified something similar, that it's a characteristic that can be an indication of regular biology or pathogenic biology, but it also needs to reflect a response to an intervention or a therapeutic intervention. So just because you can measure it is not necessarily enough. My good colleague, Dr. Carlton, and I here at University of Michigan have worked on some of this, and wanted to come up with some caveats for what we think a good biomarker is. And so it should be potentially useful for prognostication or prediction of outcome, or useful in prediction of who could potentially benefit or be harmed by a certain therapy. But then some other a perfect biomarker would be available, which we cannot say of some of the biomarkers we've studied so far in PARDS. Quick to assay, likewise potentially a problem. Reliable and reproducible and easily interpretable, and hopefully, especially for pediatrics, as least invasive as possible. These graphs here just show you that here's a whole slew of common biomarkers of inflammatory markers, and you could see ranges are so different between serum measures and bronchoalveolar lavage measures. And so the dynamic range, this is a logarithmic scale, can even be worse for some of them and quite excessive or others, depending on your source of the markers. So these are important things, especially when we're talking about the lung. And the prior speaker, Dr. Soans, so elegantly showed both the histology and the pathophysiology in schema. And in applying what he described to us, which of these aspects of alveolar injury, interstitial injury, or vascular injury can we use as potential biomarkers? Another very important thing is, when is this patient coming to you? So what part of the phases of ARDS are you measuring these biomarkers? And as the prior speaker also described, we have an edematous phase, we have a hyaline membrane phase, and again, the hyaline membranes are not the same as the hyaline membranes in neonatal IRDS. These are hyaline membranes because the alveoli disrupt in ARDS, leaking protein into the space. The water can get reabsorbed, but the protein is left packing behind. And then a fibrotic phase, which maybe not every patient will have. So where in the course, is it prior to ARDS, early in ARDS, later in ARDS, are you to know which biomarker to measure? So to qualify a little more, we'll focus in this talk for the next few minutes on inflammation, alveolar epithelial injury, vascular endothelial injury, coagulation, and these PAMPs and DAMPs real briefly. So this is a great study from Europe that described by Dr. Schouten and Dr. Bosson and colleagues' labs about the also importance of the developmental impact that from neonate to pediatric to adult. Now, I know that's common to say kids are not just little adults. That may be a little bit too simplistic because they need to evolve into adults. So we need to keep this time spectrum in comparison. And you can see for certain markers, this is myeloperoxidase, this is ICAM, this is IL-6, this is P-selectin. There may be some differences over time. And there are differences compared to normal function versus in the times of severe or other ARDS. In schema, in grand schema of early injury, early inflammation, and then later inflammation, this is work by Matt Zinter from UCSF in California, indicating that in early plasma samples within the first 24 hours of PARDS of 194 kids, you can really see a dramatic difference in the non-survivors versus survivors in the levels of each of these more common inflammatory markers. So these are all potential markers individually of prognosis. This is work, what I'll talk a lot about, expands on some of this, and is work from my colleagues, Dr. Mike Kwasny, one of my personal mentors for the last 30 years and a father of ARDS, Michael Mathey, UCSF, my current close collaborator, Mary Damer, and our colleague Anil Sapru at UCLA, related to the ancillary work we were able to do on the RESTORE treatment trial, which what the primary investigator for that was Martha Curley, currently from UPenn, related to ancillary study work we did to this acute respiratory failure study. So in this work, we really tried to see if we could identify patients who were going to have markers that could predict ARDS development. But what we found is that on the very first day of intubation, which is day 0 for acute respiratory failure, first of all, 80% of the patients who were going to have ARDS already had it, and 90% of patients who were going to have ARDS had it on day 1. And so we didn't do very well at predicting ARDS development because the vast majority already had it. We definitely saw, and this is logarithmic scale, that the vast amount of early inflammation occurs extremely early on in ARDS, and however, the marker does seem really nicely associated with PARDS versus no PARDS, and also with survival versus non-survival. And again, this is a logarithmic scale. And I wanted to point this out because this, I think, is a very fairly recent Egyptian study looking at SNP association of IL-1RA in community-acquired pneumonia. So which patients are potentially at highest risk of community-acquired pneumonia plus or minus ARDS development. And we did not find the same SNP in our work on IL-1RA, but I think more on this to come. And I wanted to toss this out there to the group because maybe some of those co-authors are on this talk right now. Going back to our work, we did the similar thing for IL-8, which is much later in the inflammatory cascade. And yes, again, we see vast increase, and this is again a log scale of IL-8 in patients who were non-survivors, very similar, and this is independent of severity of illness, all of these findings that I'm describing to you. We tried to match this then also with whether or not this inflammation can correlate with the presence or absence of bilateral disease on the X-ray, because we know the X-ray is always a conundrum in ARDS. And interestingly enough, the patients in this sub-study with bilateral infiltrates had a better clinical outcome, but we saw a logarithmic increase in IL-1RA across all days in these patients. We did not see the same in IL-8. So this is why this exists. With this, we're not 100% sure. Moving to alveolar repair. So as we talked about in the last slide, the AT2 cell is the progenitor. If they're all destroyed, they can hopefully populate AT1 cells, which might help you with fluid recovery, but then you still have a dearth of AT2 cells, so you have surfactant deficiency issues. And the jury's out. There's not that much in terms of biomarkers of early fibrosis. This is some unpublished work that we did 20 years ago, showing that precursors of fibrosis, procollagen peptide 3, on the very first day of ARDS was elevated in patients who died. So hopefully we'll be able to give you more information on this in the years to come. In terms of alveolar epithelial markers, we have SPD and we have RAGE in particular. I'll talk about that in a minute. This is work we did, again, trying to make sure always accounting for important covariates like age and severity of illness and disease associated with ARDS. And the severe PARDS patients, as expected, do have elevated SPD in the blood. And it also correlates with those who do not survive. RAGE, receptor for advanced glycation end products, is a really potentially really helpful marker of alveolar injury. This is expressed on AT1 cells, and you can see here it's really nicely not exclusively found in the lung, but it's as highly specific, and blood is sometimes easier to get than BAL in a pediatric patient, especially one who is quite acutely sick with severe ARDS. This is Nader Yaya's work, who tried to compare Ang2, which angiopoietin is a vascular endothelial injury marker, together with RAGE. Again, very early on, this average specimen time was collected within 15 hours, and both these markers are associated highly with mortality. Interestingly, the vascular endothelial marker, as expected, would correlate more with vascular-related associated ARDS, such as sepsis, which is reflected in indirect population. But RAGE, which was the alveolar epithelial marker, was highly associated with direct lung injury in non-survivors versus survivors, but across the board, not as different as you would expect. Moving to the vascular endothelium, so we have selectins. This is again published by Al-Bitagi and colleagues, again from Egypt, again showing and corroborating vascular injury highly associated with death, again, so prognostic markers. In the interest of time, I won't focus too much on the coagulation cascade, but thrombomodulin plays, as we know, an integral role in coagulation, but also in the inflammatory pathway response to sepsis. And we've seen this is Anil Sapru's work and Ben Arwol's work out of UCSF and UCLA, showing thrombomodulin in very early lung injury associated with mortality and multi-organ system dysfunction. PAMPs and DAMPs, again, this is just touching on everything in a 25-minute talk, and the prior speaker talked about this, so I'll save time by not doing that right now. But Nader Yaya did identify circulating nucleosomes, which are DAMPs, again, highly correlated very early on in pediatric ARDS with mortality. And soluble ICAM, which is a marker of macrophage activation, again, related to inflammatory process, but also a DAMP, describing, and this is again, this Egyptian group showing plasma ICAM much higher in the non-survivors for survivors, and actually corroborated work we did earlier in our group about 15 years ago. I think we will see in the next years more to come on neutrophil extracellular trap formation, or NETs, as different inflammatory processes, and 50%, as we know, of pediatric ARDS is related to acute infection, whether it's pneumonia or sepsis. But let's talk about other things that are highly relevant, and I think you want to hear a little bit about the phenotypes work. I think most people on this conference are aware of Carolyn Calfee's work from the adult ARDS, where she first described now 6 to 8 years ago in combining biomarkers and clinical risk factors into a latent class analysis, that two phenotypes exist, although the language for phenotype, subphenotype, and endotype is moving over the current discourse, but the hyperinflammatory phenotype differs dramatically from, for lack of a better term, hypoinflammatory, maybe normal inflammation phenotype. But this hyperinflammatory phenotype consistently over multiple assessments of multiple ARDS net trials has a much higher mortality and worse outcome. Pratik Sinha, now from, I think, St. Louis, Missouri, but originally at UCSF, tried to make a parsimonious model with 3 or 4 markers. Why? Because again, based on the first slide, the optimal biomarker is measurable potentially with point of care and not just in a research lab. And using the most disparate markers in adults, found a really nice ROC curve with area under the curve of 94 and 95% in the parsimonious model. So with that, Dr. Damer and I used our work from the Bali cohort for identification of latent classes in pediatric patients. And we did notice, and I showed you the individual marker results for many of these work, highly variable levels in these patients within the first 48 hours of ARDS. So we thought maybe using this plasma, we could see whether kids differ from adults that much and if they had phenotypes. Our cohort had around 328 patients, and you can see the distribution of them. Pretty sick cohort of patients, again, And we did, lo and behold, I'll cut to the last pages of the mystery novel, that we also identified 2 classes. And when you mark them out, they look starkly similar to the adult phenotypes, with a hyperinflamed phenotype and a hypoinflamed phenotype. And we tried to match the LCA done in adults as exactly as possible. And Dr. Calfee and Kevin DeLucchi were mentors with this. What I think is extremely interesting is that, as we would expect, this hyperinflamed phenotype is more seen in the severe PARDS patients, but guess what? It is also evident in mild and moderate PARDS patients as well. And indeed, the hyperinflamed phenotype had longer duration on the ventilator and a higher mortality. We also tried to match exactly Dr. Sinha's work with a parsimonious model, and we also were really thrilled and surprised to find areas under the curve exactly similar to the work seen in adults, and hoping that we can take this forward to next steps for point of care testing. So this is our first work. We don't have a validation cohort yet. We're hoping to have a validation cohort with ancillary study to the current prospect trial, which I think that Dr. Knaber will allude to later on as well in his talk. To finish our talk, I think we'll mention the gene expression analyses, which I think will also be coming forward in our biomarker assessment of these inflamed and uninflamed phenotypes. This is work in adults with sepsis-related ARDS by Dr. Boss and colleagues again in Europe, also showing a reactive phenotype similar to a hyperinflamed phenotype, and showing enriched oxidative phosphorylation, not unexpected mitochondrial cell dysfunction, and gene expression patterns related to macrophage activation. Moving back to pediatrics, because this is a pediatric ARDS seminar, Nader tried to look at PARDS subphenotypes with RNA expression patterns as well, and identified 3 cohorts, not 2, which you can see defined here. And again, these are measured within the 24 hours of PARDS onset, and certainly there's a different outcome measure for each of these PARDS categories. So this is all exploratory work. This is all in the research lab, but there's definitely going to be more to come. This is a small study related to gene expression in patients who ultimately ended up, some of them ended up on ECMO and needed advanced life support. And again, different subphenotypes being identified, and certainly the sickest of the sick who ended on ECMO definitely had a different gene expression pattern than those with MODS, no ECMO, and those of controls. One of our colleagues here at U of M, Rodney Daniels, is also looking at biomarkers related to reduction oxidation potential, and this may be particularly useful if we're starting to use antioxidant therapies in the future, like N-acetylcysteine or vitamin C, that we can potentially look at redox potential in real time to see if we can get response, use redox potential as good or bad response to our antioxidant therapies. And finally, Jocelyn Grunwell, currently at Emory in Atlanta, very recently, this is actually just a couple months old, looked at a very easy to measure airway fluid metabolites. So again, this hits a good feature for a good biomarker for pediatric acute hypoxemic respiratory failure, although 55% of her 74 patients ended up with pediatric ARDS. Also identifying 3 clusters of patients that have much different time on the ventilator in terms of ventilator-free days related to different metabolic pathways. And so in her work, and in this case, it's ventilator-free days. So ventilator-free days are the number of days that the patient is alive and off mechanical ventilation in a 28-day month. So higher is better. And so in her groups, we see all of these alanine pathways, citrulline, homoserine, methionine, and threonine pathways, much different association of outcome in these exhale breath condensates in these pediatric patients, and across a significant difference of PARDS severity of illness. So this is another potential really exciting opportunity in the future. So the goal of this is to use this in clinical trials for to predict which patients would be the best to trial in the randomized control trial, and which you don't want to include in the trial because they may be harmed. This is the whole focus of precision medicine. And I think also we want to track the biomarker trajectory over time so that you can then further tailor the treatments and the diagnostics for each patient individually. And so the holy grail for this is to do this in real time. This is again work by my colleague Erin Carlton from University of Michigan to try to establish point of care testing using microfluidics models, which are still not ready for prime time, but this would get you your inflammatory markers potentially within a 4-hour turnaround time. And Danny McCauley is currently starting a observational point of care study in ARDS, which will be prospective identification of these patients. It's called the FIND study. And this is going to take the markers found in all of those ARDS-NET predictive enrichment studies that Dr. Calfee did with the 2 phenotype group. And those were all retrospective analyses. And he's going to try to identify the similar patients and in a point of care fashion, do those same markers in real time and see if, again, in a prospective fashion, it correlates with outcomes. And then the next step from there would potentially be to use that technology in actual RCT identification of patients. And so with that, I think we may now be a little bit more close on time. And it always takes a village to do this kind of work. And this has been my village over the last 25 years of this work. And hopefully I can answer some questions, particularly at the end.